Another article type that we've been exploring is something called a microbiome announcement. So one of the challenges in this kind of work is that you gather an enormous amount of sequence data. And it can take a long time to get through all that sequence data. And in that period between first gathering the data and analyzing it and then publishing it, it can, sometimes be 2 years or more before you're actually reaching publication after gathering the data. And so we have tried an effort in a new article type called a microbiome announcement that will provide an outlet for scientists to release a data set with kind of what we call a 50,000-foot view of what they saw, not heavy on the new findings and the analysis itself, but here's an interesting data set, let's provide it to the community. We have the things that we're going to do with it, but it could be used for other things too. I mean, that's kind of I guess a good analogy would be the Human Genome Project, right? I mean, that data set, everybody working on every aspect of human health has a different slice to take out of that data set. And the same thing could be said for an environmental data set, or a microbiome data set. I'm interested in a particular group of microbes, or I'm interested in a particular biochemical transformation pathway. And so that information would be available. The bar is quite high on publishing these. We want data sets that are quite significant in scale and in kind of the diverse range of data types that are there. But we're hoping that it will be somewhat of a catalyst for attracting those excellent data sets to get them into the public realm of science, maybe a year, 2 years in advance of when they may actually make it.
Microbiome announcements they're a unique type of article at Microbiome, but also they come with, and we insist on, it's a requirement for publishing such article. It's a very detailed description of the technical and analytical tools and methods that were used to generate the data set. And I think this is essential to guarantee quality, so people can actually use the data and rely on the data in their own work.
Yeah, I would say an interesting challenge in microbiome research has been that I guess we're sort of born out of genomic research. And in genomics, there's only one piece of metadata that really matters, and that's the biological taxonomy of the organism, right? We want to know what organism did this genome belong to. For a microbiome data set, the possibilities are endless as to what this really describes. And so in addition to what Jacques pointed out with rigorous reporting of methods, is we want to also have extensive reporting of what we call environmental metadata or clinical metadata, right? So that you can place all of this sequence data into its proper context. And the utility of the sequence data increases tremendously when that metadata is more complete. So I could say my microbiome sample was from the Chesapeake Bay, or I could say it was from the Baltimore Inner Harbor after a rainstorm in January, right? Well, the difference in those kinds of pieces of data are tremendous. So that's another thing we're insisting on in the microbiome announcements.